Good afternoon and welcome to the ESHG webinar series. My name is Bill Newman. I'm the current president of the ESHG, and it's my absolute delight and pleasure to introduce this series to you. And thank you to all of you for joining us today. We have put a lot of thought in the ESHG into some of the resources that are available that we provide in terms of education and training and learning opportunities for our members. And of course, you know about our wonderful conference that we have every year in the summer, but a number of other resources that are available through the education committee's work, through face-to-face courses, some of those in Bertinoro in Italy, but in other centres around Europe on a number of different topics. We've had a podcast, we've had app-based learning on neurogenetics, and we've been seeking ways of trying to increase access to some of these opportunities by recording them and making some of the major talks from the ESHG available from the conference, but also for the last few years since COVID, having turned to a hybrid type of conference. And so one of the things that we felt there was an opportunity to do was to start a webinar series. And particularly this gives an opportunity to some of our wonderful scientists and doctors within the society who have been members of the Scientific Programme Committee, who, as you may know, when they sit on the Scientific Programme Committee, aren't eligible to present their work at the Society's meetings. And there are lots of reasons for that, to ensure that everybody gets access to opportunities to present their work, and to ensure that that's as fair as possible. But it means that for a few years, some of our great people don't get an opportunity to present their work. And so we thought this might be a nice forum to do that. And so we've been thinking about some of the topics that people would find interesting and looking and approaching some speakers that we felt have been doing some wonderful work and would be of interest to our society. So it's my absolute pleasure today to introduce to you our inaugural speaker for this ESHG webinar series, who's Dr. Malte Spielmann. Malte is a fabulous pediatric geneticist who has done a wonderful research initially with Stefan Munlos in Berlin, where he did lots of work around TADS and chromosome architecture. And then he moved to Seattle to work with Jay Shenjur, and more recently has moved back over the last few years to Germany, where he's taken on position of professor and director of the Department of Human Genetics at the University Hospital Schleswig-Holstein in Lübeck and the University of Kiel. So today, Malte is going to speak on a topic which I think will be of great interest to all of us: the dark side of the genome, challenges and opportunities in the post-genomic era. If you have questions for Malte throughout his talk, please post those in the Q&A button, which you'll find on your screen, and Malte will field questions at the end of the talk. Thank you so much.
Yes, thank you so much, Bill. This has been a very great introduction. I'm just trying to share my screen here because someone said they couldn't hear us. Not sure if that's true. I will just try again. So if you can please let me know whether you can hear me and see my screen, then I will start.
I can hear you perfectly, Malte.
Perfect. That's great. So then, so it is my enormous pleasure to be here, and I want to talk to you about something that sounds mysterious, but I think is clinically extremely relevant. And we call it the dark side of the genome, but most people talk about the non-coding genome. And just hang on, I have to get rid of something here. So perfect. I want to start talking about my clinical work. And when I was a clinical geneticist in training, I always had a very hard time to make sense of all those rare diseases. And I know there's many people of you out there who can basically look at the nose of one of these individuals and know exactly which disease it is. But for me, this has always been very difficult. So I always dreamed of a technology that could do the trick for me. One test that could solve all my problems. And then almost 10 years, now 11 years ago, this paper was published by my dear friend Christian Gielissen and the entire team from Nijmegen. And it was the first paper that showed that if you use whole genome sequencing, you can basically solve all the different kind of genomic rearrangements, single nucleotides, copy numbers with just one test. And they were able to solve almost 60% of all cases with severe intellectual disability. This has, to be honest, never been repeated, but they were first, and we are still trying to get there. So many years later, I became head of the Department of Human Genetics in Schleswig-Holstein, which is the most northern state in Germany. And we decided to do whole genome sequencing, just like Christian and his friends had done many years earlier. And now since 2022, we do this as a routine test. And I want to show you some of the results and then maybe discuss with you why that's still not enough. So I think if you're talking about whole genome sequencing, then you're really talking about the ultimate Swiss Army knife. I'm talking now about short read, but we can talk about long read later. But if you use it correctly, you are able to get almost everything out of the genome there is. Obviously, you get the sequence, but then obviously there's also, you can reconstruct the chromosomes, but also deletions, and more recently also repeats. So that should do the trick. When we did this for 2 consecutive years. We were able to solve roughly 50%. So I want to stop here and say this is a great success. But when I talk to some of the parents, they say, so what's wrong with you? You sequence in the entire genome and you only get half of them right? And I must say this, when I say 50%, this includes also the VUS, right? So I got pretty shocked and I thought maybe we're just doing a very bad job. But when we looked into the literature and looked into the greatest kingdom of all, the 100,000 Genomes Project also only got 35%. So we were even a bit better. So still the question is, if you talk to a patient or a parent, they say, what are you missing? And this is the question that I want to ask today. I mean, I think we're doing pretty good. We always throw the newest technology at our patients, but diagnose-wise, there's still lots of room to improvement. So what are we missing? And I would say the questions here, and I'm throughout my talk, the next 45 minutes, I want to just ask this question a couple of times and introduce different ways, I think, of what we could do better. So I think the first and most obvious thing is that we're not looking in the right places. If we look at the genome, and every time I show this slide, I still think it's a scandal. Only 2% of our genome actually has genes, and 98% has non-coding sequence and used to be called junk DNA. And I don't really like the term junk because it's a bit offensive, but I much prefer the term gene deserts, right? That kind of implies that there's something to discover. But that also means that there's lots of sand in those deserts and you can drown or die of thirst because you might not find that pyramid or that hidden tomb. But that means we only understand and focus on an entire attention of just 2%. And then what do we know about the non-coding genome? And not so much, but let me tell you a little bit about what we know. So your 2 meters of DNA are tightly packed into the nucleus of each cell, and not randomly, but there's actually certain territories. And then if you zoom in further, then you see that there's active and inactive compartments. And if you zoom in even further, and that's what I want to talk about today, you see that there's genes, and then there's non-coding elements called enhancers. And because and these enhancers are kind of like light switches. They turn in the genes on in the regions where they should be. And then when you look at this in more detail, you see that there's two different regions here. So since 2012, we know that there's not only genes, but also regulatory domains. So let me walk you through this. What you see here is one gene expressed in the embryonic limb, and it has several enhancers, and those enhancers keep looping towards their target gene. And here you have a gene expressed in the brain that also has enhancers expressing it in the brain. But then up here, what is a high C map, you see that there's lots of looping going on. And when I first saw this, I thought, what the hell does it have to do with my patients? But then I thought, what about those patients that don't have a deletion in the middle of nowhere, or that where I don't find a mutation? So then I thought, how does this enhancer know that it's only supposed to drive that gene and not this one? And these boundary elements here, they seem to keep them apart. So I wasn't just thinking of any patient, but a few years before that, I had seen several families with a disease called brachydactyly, so short fingers. And we had tried everything. The families had deletions of a gene called FRNA4, but the gene was not related to the disease. The knockout mouse didn't have any phenotype. But now, with the knowledge of those regulatory domains, or TADs, we try to understand it, and we thought maybe the deletion actually removes the boundary here. So we came up with an hypothesis, and we thought in the wild type, here you have two TADs, one expressing a gene in the brain, one expressing in the limb. Now, if you remove that boundary, what would happen is that the boundary is gone. This enhancer here can hijack the gene and drive misexpression in the embryonic limb. And it took us lots of mouse experiments to prove our point. This was work that was still done in Stefan Mundlos' lab, led by Dario Lopianis. But to our surprise, we could show that this is indeed a disease mechanism, and we termed it an enhancer adoption or enhancer hijacking. Basically, this enhancer hijacking the neighboring gene. But what was even better, that over the years, then, we found lots of other disease where this mechanism of either fusing a regulatory domain or even creating new regulatory domains called neotads, or just removing individual enhancers could cause disease. And we showed this mainly in congenital disease, but it all turned out that this was also a disease mechanism in cancer. So while in the beginning people were laughing at us, then large journals actually asked us to be on the cover. So we were extremely proud. But more importantly, what was great, that in the cancer field, this mutational mechanism was basically discovered in parallel. For example, here in medulloblastoma, they termed it enhancer hijacking. This is a very frequent cause now of disease of medulloblastoma, and also in certain leukemias, they called it chromosome neighborhoods. So I could stop here and say this is all a great success, but obviously it doesn't stop there. So I think we were quite okay at establishing this as a new disease mechanism, but is it going to explain the other 50%? I'm not sure. So I have to ask a question again. What else are we missing? And maybe we are simply overlooking it. And I think this happens a lot, that you see it, it's right in front of your eyes, but you don't realize that this is actually a disease-causing mutation. And I want to tell you a next story that once again has something to do with a non-coding genome. And I want to introduce Martin Menzer, a very clever physician scientist who worked with me in Berlin. And many years ago, we had this patient sent to us from Hong Kong, and we did whole genome sequencing at the time. It was very expensive, and found a frame shift mutation in a gene called HMGB1. The problem was this gene was already taken for mild intellectual disability. So Martin and I didn't know what to do. So we discarded it. We just put it in the drawer and said, we don't understand it. But then I moved to Schleswig-Holstein, and one of the first patients that so this is basically what it looked like. So we had mutations in that gene cause mild intellectual disability, frame shift, but also missense. But we found mutations in the very last exon, and it didn't make any sense, so we put it away. But then when I moved to Kiel, the very first patient that was presented to us was this guy, and Almut Kalibe, the clinical geneticist working with me in Kiel, had seen her prenatally, had seen that patient prenatally, and said, well, this has very similar abnormalities to your patient. Basically, it has abnormalities in the hands, it has a polydactyly, and it has no tibia. So then another very clever clinical geneticist from Berlin said, this is not just some random syndrome, but roughly 7 or 8 patients have been described worldwide, and it's called brachyphalange polydactyly and tibial aplasia syndrome. So we thought, well, let's just do whole genome sequencing and we're going to solve the case. And we did that, and to our surprise, we identified the very same de novo frameshift mutation in the last exon of this gene HMGB1. And once again, this doesn't make sense because those patients shouldn't be so syndromic. They usually have just a mild intellectual disability. So I went back into my high school, or basically first year medical school, biochemistry knowledge, and thought, well, what about those frameshift in the last exon? Sometimes they create actually a stable protein. So we took a closer look, and indeed, usually the protein stops here, but these mutations in an intrinsically disorganized region, so a region we would usually discard, actually caused a new tail, and this highly acidic tail, and made a new protein. So we thought that could be interesting. And since this is a transcription factor, I wondered how could that be related to genome architecture? And I asked my dear friend Dennis Shins, who is in the lab right next to us. And I told you earlier on that this chromatin folding, the contact between enhancers and genes, can be measured. And I always envisioned it like this. So you have an enhancer and then a transcription factor driving gene expression. But when I talked to Dennis about it, he says, Malte, you are completely naive. This is not one transcription factor, but it looks more like this. There's many, so it's actually really it's a hub. There's many. So what would now happen if you have a mutation in this acidic tail? And our idea was maybe this changes the conformation of this enhancer hub. And so one of the clever PhD students of ours then, of Danish, made an assay, and we used HMGB1, our gene, and just put a GFP on it. And when you put that into wild-type cells, you see that HMGB1 goes into the nucleus, and because there's a bit of green in there. So this process happens very fast. You don't see anything, really. But what would happen now if you have your mutation? Then our idea was this would get more sticky, and you see internuclear condensates. And this is exactly what we could see. In our mutant, suddenly we see those condensates, and transcriptionally, they go insane. These in vitro, these cells actually die. And some of the reviewers actually asked, how do these patients live? But we thought that this is a new mutational mechanism, and this process is actually called phase separation. So we sent it to a journal, but they said, oh, we don't care. It's so rare. You have those 6 or 7 patients. So then Danish and I decided, let's try to go bigger. And this hardly ever works, but we tried to look and see if this is a more common mutational mechanism beyond just rare disease. And we downloaded lots of proteins, or lots of from the databases where you have a disordered tail. And then we looked for genes that have a frameshift in the last exon in some of the common databases like ClinVar. And we were able to identify over 600 of those mutations in different genes. And then we thought, well, let's try it out. Let's try our assay and see if this is actually a common disease mechanism. And I will not show you all of them, but for example, here, HMGB3, very closely connected to HMGB1. Same thing in the wild type, it's a little bit green, and in the mutant, you see those condensates causing disease. And we can go through all of them, but let's focus on this one here, RUNX1, a very frequent cancer mutated gene. The same thing is true. In the wild type, you have a bit of condensates, but they get much more severe in the mutant. So we believe that this is really a new disease mechanism. When I told this to my colleague Nadine Hornig, who works on completely different disease called differences of sex development, so basically individuals who are in between male and female. And she usually this is caused by mutations in the androgen receptor. And back to back with our paper, she and my institute was able to identify mutations in a completely unrelated gene called DUM2, but DUM2 actually forms
condensates with the androgen receptor, and you can visualize them using this cool technology. And when you have mutations in DAM2, these condensates don't form anymore. So in this case, it's the opposite. It's not you have too much condensates, but those condensates don't form anymore. And you have a loss of condensation and thereby a loss of androgen receptor function. So the kind of the same mutational mechanism, but the opposite. Once you have too much condensates, and here you have not enough. We also quantified this, but I will not go into the details. So basically what I tried to tell you here is we believe that chromatin folding is not only affected by large structural variants, but you can also have coding mutations that then again, genome-wide change chromosome architecture. Good. So I told you now two things that I think we are missing, but that's still not going to do the trick, right? Maybe that's another 2% or 3%. So what else are we missing? And maybe, and I think we can all agree here, we are a technology-friendly society. Maybe we are using the wrong technologies. And here I want to cite, or basically just show the paper that everyone always shows. I usually show this to my medical students and say, well, we thought that we had the whole genome sequence going, but every other year, and now finally in 2022, they actually filled the gaps using long-read sequencing, so T to T, including different technologies. In our lab, we mainly use PacBio hi-fi sequencing, but I'm sure many of you use nanopore. I think it doesn't really matter. But what the difference is that you actually get the full rearrangement spectrum using this great technology. And with PacBio hi-fi, you actually have Illumina accuracy. So I wanted to really use this technology. And I want to give you one example that I think was quite striking to us. This is from our institute, and Jörg Hellenbroich is the attending and head of clinical genetics in Lübeck. And 21 years ago, he described a family with a disease called spinocerebellar ataxia type 4, SCA4. And this is a large family by German standards, and they were all affected. He mapped them, and he found a locus, mapped a locus on chromosome 16. So, but then we tried everything, literally everything. They Sanger sequenced all the genes. Then they did short read sequencing. They did exome sequencing, and we couldn't find anything. And then I told them when I came, let's try PacBio. And we used PacBio on just 2 individuals and were able to identify with the first one, sequence a coding repeat in a gene called ZFHX3. And I couldn't believe it. And I asked them, please confirm it. And we had a very hard time confirming it using any other technology. In the end, what happened was that that very same week, when I was so proud that finally we could do long-read sequencing, I invited someone from Tübingen, which is the largest institute in Germany. And my good friend Tobias Haag came along, and I told him, you know what, in my institute, we do long-read sequencing, and we even found something in a cool family. And then he said, but you don't mean SCA4, right? And I was like, oh Jesus. And so I called up my friends, and they said, no, it's indeed SCA4. And they had found the very same repeat in the very same week. And we found out later that 2 other groups worldwide found it also. So it really shows that, and this is disease causing, I'm not going to go into the details, but it was basically invisible using any other technology. And it also shows that if you collaborate, so Tobias and I and many others paired up, and we were able to publish a beautiful paper last year. So I think there's more. I'm not entirely sure how much more, but I think we can discuss it in the end of my lecture. But I'm pretty sure that this is going to be exciting in the future. So now, summarizing again, what did I tell you? We are currently missing the structural variants in 3D genome. We are missing the chromosome modifiers. And now also the long-read sequencing technology will give us more. Now I want to come to my last part. And maybe we are simply using the wrong approaches. And what do I mean by that? So I think the main problem is that we can do next generation genotyping now. We can sequence anyone here in this call. I think there's now a lot of people in the call, 299, that's a lot. But sequencing wise, that would not be a problem. And then we could even use CRISPR to make all those mutations. But then how would we analyze them? And that's the biggest challenge, I think. We are very stuck still in the 80s. So making one gene knockout and then a cell line, or one mouse and then analyzing it for an entire PhD. So I think we need next generation phenotyping strategies. We need to understand disease at higher scale. And how could we do that? And I want to tell you one story that led into a whole series of more stories. So in my lab, we do a lot of mouse work. And what I'm showing you here is the organogenesis of the mouse. And why is that interesting? Because most of the disease we study in congenital disease happen during organogenesis. In humans, this takes 4 weeks. In mouse, only these 5 days. And you see here from day 9.5, the mouse is basically just a brain and a heart. And then till day 13.5, all the internal and external organs are formed. But what I'm staining here is just one single gene. But I want to stain all the genes. So I was wondering, could we develop a technology where we don't have to kill one mouse after another, but we build a digital mouse. So we take all those cells, ideally at single cell level, and then reconstruct the mouse and on the computer. And then we could do all our tests online. And how did we do this? Back in 2019, my colleague Jun Kao and I developed a single cell sequencing technology called SKY. And I will not talk about it, but basically we could sequence millions of cells. And by that, build a digital mouse catalog of all the different cell types. And what first looked to me like modern art is actually all the different cell types that a mouse has. And here you see, for example, all the endothelial cells, and these are the liver cells. But still, that doesn't tell me where my disease is or where exactly my favorite cell types is. So I told you now that we built a cool technology that can take things apart. And usually you hear people talk about a smoothie, but I like this example better. So what single cell sequencing or our technology can do is you have something beautiful like this car, and now you can take it into 1,000 pieces, right? Like this. And I think then you have to understand the biology. And for example, this piece here, the tire, we can all agree what that does. But then suddenly it gets very difficult. So here is the steering wheel, right? That's still easy. But what about all those tiny pieces? And that's exactly what happened to us in the mouse. We identified some cell types that we thought were obvious, but so many that we weren't even sure if they were real. So we needed better algorithms to understand a mouse development and then to study disease. So we paired up with Cole Trepnell, who is a god of single-cell bioinformatics. And he had a technology, algorithm called Monocle 2. So we wrote a new version called Monocle 3. And that you can now use to follow your favorite cell types through developmental time. And then ideally you can see, well, this is the transcription factor, or this is the stimulus from outside that makes your cell go into that, your cell type go into that direction or this direction, or into health or disease. And how does that look like? I will just give you one example. So let's imagine we want to study embryonic muscle development. Usually you would have to slice up a mouse, and a PhD would be very busy for many years. But here you could just tell the computer, show me muscle development. And then it will show you that the dermatomyotome here, the side, will then develop into 2 distinct ways into mature muscle. And then based on marker genes, you can identify that these are indeed muscles. And when I first saw this, I thought I'm going to win the Nobel Prize, because there's 2 ways to make muscle. But a quick literature search or just a Google search told me that this had been known for many years, but it took them 10 years and many mice needed to be killed. So now we can do this on the computer. So this just shows you how powerful such a technology can be. And this wasn't enough. We wanted to actually build a real digital embryo. This is still work together with Jay Shanduri's lab. And so a very clever PhD student in Jay's lab, CX, decided, so couldn't we just make a mouse from the zygote stage till birth, basically? And he downloaded all the data from the internet he could get. That was only till halfway through, 15.5. And then he sent this to me. And I was surprised, and I said, CX, so you've been to London maybe and taken the tube? But when I took a closer look, I found out that indeed this is actually embryonic development using single cell analysis. And here you see this is with the zygote stage. And here we highlight every single transcription factor that is necessary to make a certain cell type. For example, here you take that cell fate decision into epidermis, or down here into other cell types, other epithelial cell types. And now we can really reconstruct mouse development, and in the future, hopefully also mutate these. So we actually don't have to kill the mice. We can first do this in silico and then do the experiment afterwards. This wasn't enough for us. So then Jay really wanted to do with my lab an experiment where we profile mouse embryonic development every 6 hours. Since we couldn't do that, we paired up with the Jackson lab, which is the biggest provider of mouse samples in the world, and they provided these mice every 6 hours. And we then together did the large experiment. And what you can see here, we really made a movie that you can now watch online. It was published earlier last year. And you can look at your favorite cell type in the embryonic development. And because this has all been published, I'm not going to show you all the data. I just wanted to show you one example where we were able to identify something really cool. So if you imagine what's the most stressful experiments of the most stressful time during embryonic development, and it is obviously birth. And this is exactly what our data show. Here I'm showing you a plot. This is the cell types before birth, so at 18.5. And then you see, for example, in the liver, the cells cluster on this side, also in the adipocytes, so in the fat, and in the lung. And then something magical happens. Around the time of birth, these cell types completely change their identity. And when the mouse is born at P0, suddenly there's no overlap whatsoever anymore in all those cell types. So when I first saw this, I was like, oh my God. So this is new. We always thought that this was the case, but we've never actually seen it or measured it. So for the lung, I thought it's obvious because these mice start breathing. So maybe that's the difference. And for the hepatocytes, I thought maybe they start drinking milk, right? And that's why this changes. So we did one experiment that sounds very brutal, but it was very important. So we took a pregnant mouse at 18.75, so just immediately before birth. And then we let her give birth to 2 or 3 pups. And then we sacrificed the mouse. And then we did the same experiment every 20 minutes. So here you can see the experimental setup. The first 3 pups were birthed naturally, but then these never took a breath. And we just let them see how fast those changes would occur. And this is what we see. No matter what happens, even if they don't breathe, just being alive makes this transition of cell types happen. So basically being on this earth is a new factor. And I'm not telling you that I've completely understood this. I'm just telling you that this technology of phenotyping is now so fine that we can see phenotypes that we were never sure we could ever visualize. With this, I would like to come back to my actual topic and talk how could we use this to study disease. So I should talk to you now about non-coding. I talked about you that I think single cell sequencing is a cool technology. How could we now use it to study disease that are relevant to human geneticists? And with this, I would like to come to my favorite topic, and I call it single cell human genetics. And here the idea is that if you have a syndromic patient, the problem always is that every single cell of this patient with Cornelia de Lange syndrome has a mutation. Yet every single organ seems to react very differently, and we call this pleiotropic effects. And normally what we do is we just focus on our favorite organ system, right? If you're an eye geneticist, you focus on the eye and you publish it. If you're a heart geneticist, you publish the heart. So we wanted to study the whole embryo, the whole animal, and this can be done with this technology. So what we did, I was very lucky that Jana Henk, my PhD student, and Fanny, another PhD student I share with Jay, were able to go onto the suicide mission where we said, let's push our technology. We wanted to investigate not one embryo, but 100. And just to bear in mind, usually an experiment like this would take one mouse, would take probably 1 PhD. So, but these brave guys actually did it. And we wanted to build a digital mouse clinic. So what they did is they chose over 100 embryos, some very severe and some very mild. And then they did single cell RNA sequencing, the way I said it before, and generated a large map of cell types. And now the real work started. We have now all this digital data. How do we see our phenotypes? And for this, Fanny was able to make a new score that was very helpful. And we called it Loch Ness in honor of the beautiful monster living up in the UK or in Scotland. But the idea here is that because we have wild type cells and mutant cells, and we can investigate them on the computer together. So if there's no phenotype, we have a mutant score, Loch Ness score of 0. But if there's more mutant cells, we call it mutant high or Loch Ness high. If there's no mutant cells, we call it mutant low. And because this is difficult to understand, we thought, let's make it easy so any clinician like me can understand this. So now you can visualize your mouse mutant on your computer. Basically, going to the right means gain, left means. So let's do that with our 100 mice. And we see here that, for example, this mouse here, TTC21B, looks pretty boring. It's in the middle. Some others have some losses and some have a weird shape. So now, without killing any more animals, we can zoom in further. And let's look at GLI2. Up here also looks pretty boring. If we now look into the different cell types, once again, no killing animals anymore. We just look at it, all looks pretty boring. But then down here, there's something interesting. There seems to be a bimodal distribution or like a shift here in the floor plate and the roof plate. And this is a well-characterized mutant, but this had never been described. But we can see it quite clearly here in our mutant score. So now we had to go into the brutal work and actually prove this. So it took us just a few clicks to generate this map, but then to actually prove our point using good old-fashioned technology, we could show that indeed these cells were actually cells from the roof plate that had now moved into plexus choroideus, where they shouldn't be in the mutant compared to the wild type. So once again, showing you that what this technology can really do. And I don't have time to show you all the data, but I think what is really cool about this is that now you can take a pleiotropic mutant like this one here. SOX9 is a gene almost expressed everywhere. And we had published this many years ago just with a digit phenotype because we only care about hands. But in our paper now, we could show that there's actually a phenotype in the brain, in the heart, and also in the liver that had not been described before. So now I showed you how you can use this technology to study disease, but how could this also help our patients? And for this, a very clever PhD student of mine asked me, well, we do a lot of single cell sequencing. Couldn't we use this for your patients? And I said, sure. So she should ask me, how do you actually do diagnostics? And please take a look up here. I said, well, we have a patient with a limb malformation and we sequence it, right? And if it has a mutation, or he or she, in a known gene, we make a diagnosis. And then she asked, what about the genes that you don't know anything about? And I said, well, we cannot make a diagnosis. We don't know anything about. And then she said, and sometimes we do expression analysis in situ or so. But then she said, well, but we now have all the single cell data. We could just use them and write a machine learning algorithm to help you make diagnoses. And I said, okay, let's do it. And this is exactly what she did. And we called it stigma. And the idea is, let's say you have a limb malformation, but you can use any single cell data from the internet, and the entire human body has been sequenced. And you upload limb data. So this is what we did here. And then you generate a map like this. This is the different cell types of the limb. And then we used a very simple approach that we call guilty by association. And what does that mean? It means, well, we know that there are certain genes causing limb malformation, like GLI3. So here in green, so we can show where they're expressed in which cell type and where in developmental time. So now tell me, computer, where are genes that are exactly like my disease genes? And why is it called guilty by association? Because the idea is not that these are really disease genes, but the idea is if all your friends are bad guys, it doesn't prove you're a bad guy, but it makes it much more likely, right? So that's why we thought, let's use this approach and find those guys who are really close to all the bad guys. And then we still use other algorithms to prioritize it. And then we can find new disease genes. And in this study, we were actually able to identify 34 new disease genes in congenital heart disease. Check it out. It's all online. But I'm free to also help. We are just expanding this data set for stigma and to make it applicable also for limb disease and brain disease. So it will be available soonish. With this, I want to come to an end. I have 4 more minutes or 5 more minutes. And I want to talk to you, what are the next challenges? And there I want to come back to the non-coding genome. So what I call the dark side of the genome is depicted here. And I stole this from a colleague of mine, the idea to depict it like this, Nadia Imke from Berlin. But I really, really like it. So the idea is, if you're an alien and you come to Earth, and this is how human geneticists have approached the genome. So then you obviously go to the places where there's lots of light, right? The genes. That's where most of the magic is. But we all agree that there's also something in between those lights. And now, in the recent year, this has been extremely exciting in that respect. For example, in the middle of nowhere in Greenland up here, not only did my dear friend Nikki Whiffen identify the most common cause of intellectual disability are now RNU4 mutations in a non-coding RNA, back to back with the other team. And we all didn't see it coming. And at the same time, Donald Trump also identified that Greenland is exciting. So I'm not sure how this is connected, but at least something is going on in the middle of nowhere. And then also coming to therapy. I think the non-coding genome is the next frontier also for therapy. Because if you look at sickle cell anemia, the only CRISPR gene therapy approved so far doesn't target any of the genes, but one of the enhancers. So I think our main challenge should be to focus on the non-coding genome. And how should we do this? The main problem here is that 90% of our mutations in our genome fall into the non-coding space, and we don't understand it. And there's so many of them. So I think what we need to do is, I told you we can do next generation genotyping, we can do next generation genome editing, and now we can even do next generation phenotyping by single cell. Now we have to combine them. And this is what I call personalized genomics. So we can now build in one cell dish, we can, with CRISPR, make a different mutation in every single cell and then have a single cell readout. And by that multiplex also our functional testing. So we can really build next generation functional genomics. So let me show you what I mean by this. And this is in the last 3 minutes. Could we test thousands of variants in a single experiment? And this is unpublished stuff. So the idea was, where do we get lots of unpublished data from? From GWAS hits. And I know you're not all interested in GWAS, but some of you are. And the main idea in GWAS is that you have a lead SNP and many other SNPs who are also significant, but we don't know which one is the disease-causing SNP. And we wanted to find out, couldn't there be a high-throughput way to test this? And we focused on cardiovascular disease because there's over 250 GWAS hits. So we thought, could we test all of them in one single assay? And once again, I was able to persuade a PhD student, Veronica, in my lab to use CRISPR screening, where you make a different guide in each different cell and then see what the transcription of the nearby genes, how it is affected if you target the enhancers, if you target the GWAS hits. And this is exactly what she did. So in the current experiment that is hot off the press, she targeted over 265 loci in different cell types, monocyte and endothelial cells. And obviously, you need a lot of controls as well. And then made roughly 3 guides per GWAS hit. And this resulted in over 1,000 experiments in a single dish. And then you run them through the experiment. We use a lentivirus. And in the end, what you get is a plot like this. So I'm just showing you one single data point. This is the GWAS hit that we targeted. And we were surprised to see that this individual hit has gene expression changes not only on the neighboring gene, like people usually assume, but also on genes very far away, up to 400 kb. And you can now, the cool thing is with single cell expression, you can also measure this and quantify this at a genome-wide level. Here's another version. This is another GWAS hit. And you see here that the nearby gene is affected, but also the next gene is affected. And once again, we can quantify this. Why do I think this is important? Because I think we can generate lots of data and finally understand the genome architecture of the non-coding genome. So why do we need so much data? Because we want to not do the experiments in the end, but do this computationally. But the computer guys need lots of data. And with an experiment like this, you can generate a large amount of data in a single experiment. And I actually think you could even personalize this on a clinical level, but we can have a discussion about this afterwards. So with this, I want to show you what I think is next. I think what is happening right now is that people are generating entire organismal maps of mice, but also of humans. And we will be able to build a cell oracle and maybe even a disease oracle where we don't have to do all the experiments anymore, but then be able to predict it. And while we are speaking here, people are actually doing this. I hope you've all seen this paper, at least in bacteria. This already works. This is the cover of Science magazine in November 2024, where people have successfully done this using a foundational model, so the biggest large language model. And I think we are almost there. But for this, we need to work together, because only in large quantities of data we will be able to do this. With this, I hope I could convince you with my brief talk today that genome sequencing is not enough. We have to go beyond just the coding sequence and probably beyond short reads. And I think this is most dearest to me. We need next generation functional genomics. We cannot just do it one by one or one lab at a time. We need to do this together at large scale. And at the same time, I hope I could convince you the future is bright and exciting. I even forgot an D here because I was so excited about this. With this, I have to confess to you that I didn't do any of the work. This was done by my lab and by some collaborators. I want to thank them and also the funding agencies. I want to thank my clinical team. And I want to just emphasize here that if you liked what you saw, if someone of you out there is interested in working with us, this is our new research building in Lübeck. And if you take a very careful look here, then you can see that there's still an empty bench or an empty computer that you could work on. So please write me an email if you would like to work with us. I'd be happy to talk to you. And now I have the great privilege to take questions. But before I take questions, I would just want to emphasize this is the first of a starting series of talks like this. And the coolest guys in human genetics are going to be speaking. And I can't wait to hear Nikki Whiffen talk about the non-coding RNAs that she's recently identified on the 26th of March here in the same location. So please come and check her out. With this, I will close my sharing and take some questions from you. And please put some questions in the chat, and I will start here. Good. So Elisa Benetti is asking, what tools and annotations do you use in your lab to analyze deep intronic variants and structure variants that fall into intergenic regions? Also, how do you prioritize these variants? So that's a great question. And the answer is it's not a trivial one. So I think I would start from the clinical perspective. Don't go into the abyss, but start with a clinical smoking gun. If you have a candidate gene and you have a mutation or an intronic variant in that candidate gene that is usually causes the phenotype, then those are the ones you should go after. Don't go for the non-coding variants in the middle of nowhere. But then how do you go on? I still use the UCSC browser. Maybe that's outdated, but in the newest version of the UCSC browser, it shows you the tags. It shows you most of the enhancer data available. And this very quickly gives you lots of information that will be able to give you an informed idea of, does it make sense to go deeper into this case? Now, this will just give you an overview, and then in the end, it still takes some brutal work, experimental work, to actually make a diagnosis. But are there computational ways to do this? And yes, there are. So we are currently writing a review where we try to put this all together. But one that I really like, and I'm not trying to advertise anything here, is called Postre. It was published in Nucleic Acid Research, and there you can upload basically your CNV, and it will computationally put all together all the data that are online available and then prioritize your CNVs. This requires a bit of computational, it has a shiny app, but the better version goes through the command line. But there's several other tools. And another option that I think very important is we can do this, we have to do this together. So reach out. I get lots of emails like this, and I can take a quick look, or I can, for example, if something in the eye is happening, then I usually hand it over to Elfriede de Beer, who's an expert there, so we can do this together. Now, Amjad Tanoli asked a question, does the boundary has impact on enhancers? Means how in absence of a boundary, an enhancer binds to multiple promoters and transcribe genes. And the answer is complicated. So I made it sound very easy and said there's just the boundaries, and usually these are associated with CTCF. But indeed, we know now that if you delete CTCF genome-wide, the boundaries almost disappear, yet the TAD structure kind of stays similar. So it seems to be more than just the CTCF site or just CTCF binding. But so the idea is that the CTCF does not only bind to boundaries, but it also binds to enhancers and facilitates this looping mechanisms. And you also have CTCF bindings at the promoters of genes. So indeed, I would call it more a structural protein, and the boundaries are just part of the puzzle. And what we've seen, so I showed some examples, and we've done lots of work on this, is that if you remove an entire boundary region, then you, like with a large structural variant, then you get enhancer hijacking. But if you just mutate a single CTCF site, usually this does not happen because the genome seems to be pretty robust. And then the cryptic CTCF site or a hidden CTCF site can basically take up the function. So then the next question from Simone is, so a localized change can start a domino effect also very far away? And the answer is yes. So if you remove, in the studies that I showed, if you remove a large TAD boundary by, let's say, a big deletion, indeed, all the genes in neighboring TAD can be misregulated. And we've seen, weirdly enough, that, for example, these enhancers, they are evil guys. They don't care what they drive, and they can misregulate several genes in a row. We've also seen every once in a while that for some reason, a certain gene or promoter seems to ignore the enhancer, and it just jumps to the next one and misregulates it. But the basic idea here is that the enhancer misregulation has some effect, usually during development and in cancer. And some genes just get misregulated, but nothing really happens. So you, once again, I come back to the smoking gun. If you want to diagnose a patient, don't start at the deletions in the middle of nowhere or some TAD boundary. But if you have a deletion of a TAD boundary next to your favorite gene that you know well, or that is the known disease-causing gene, those are the ones we should go after. Jackie Beckman asked, need work regarding your personalized genomics. Can you also challenge cell-cell communications, may given different results than looking on isolated cell types? And the answer is yes. I mean, I once again made it much easier than it actually is if you would take such a personalized genomics approach. And I just want to highlight what I mean by that. I think I think I made clear that I don't think it makes sense to just go one mutation at a time. So now we have to compromise, and we could, in theory, take all the variants from an individual patient, let's say a rare disease patient or even a cancer patient, and put them into an assay like this and test all of them, and then focus on the very few ones that actually show a significant result. And this would be kind of a screening, a personalized screening. Is that going to be enough, or is there, because you have to compromise. You can do this only in one cell type, or maybe an immortalized cell type. But I think it's going to at least start somewhere. But could we do this also in a more complex system with many cell types? I think the answer is yes. In an organoid system, for example, or my lab is also working on this to do this in vivo, to actually in mice, that you use several inserts into an embryonic stem cell and then investigate them once the mice are growing, right? This gets challenging and in the end also quite expensive at the moment, but I think it's just a problem of time, right? So I think this is going to be exciting. So, and Nadide Randa is asking a question. I also want to ask a question about the mutations in the last exon of a gene. How can we apply this to the criteria of the American College of Medical Genetics? So this is going to be difficult, I agree, but I think the main take-home message here is that we have to understand that, and many people do this already, that one gene can have many different phenotypes, right? And the haploinsufficiency can just be one disease mechanism. If the last exon is affected, for example, then it can have a very different disease mechanism. When I first presented this work, when it was still unpublished, I went to Bertinoro, and I really like presenting unpublished stuff because it sometimes makes your idea better, or it shapes the idea, and it also invites people to give positive criticism. And Han Brunner was in the audience, and Alex Heuschen, and they said, well, we have this disease and we've worked on this for a long time, and they call it's Robinot syndrome. And Robinot it's a skeletal dysplasia, and the gene is DLV1 has mutations in Genome all over the place, but only the last exon ones, the frameshift ones, are caused this Robinot. And we tested it in our assay, and it showed exactly that phenotype. So I'm not sure how you can include it into ACMG criteria. I think you just need to think about it. And that's what makes an academic institution that then you can take such a case and say, well, maybe it doesn't fulfill the criteria yet, but we could run a small assay or contact our collaborating lab, for example, mine, but also many others, and make it pathogenic by that. I think this is going to be okay. Okay, now there's a completely different question, and I would like to take the opportunity also to take that. So this is Lisa Roberts coming in from South Africa. So hello to very far away. I'm glad you are here. And she says it was a nice talk, but what's the best tip for those about to embark on genome sequencing for the first time? What did you wish you knew when you started? And I think this is an important point. And I think I would address this from a really practical point of view. So we all know that the Broad Institute can do it. We all know that the Sanger Institute can do it. But how does that help me in my place, right? In my lab, in my institute, in my hospital? And that's why when I moved to Schleswig-Holstein, we were in the same position. We knew that the big guys can do it, but how could we do it? And I think the biggest challenge is obviously bioinformatics. And so we actually paired up with a commercial partner for our clinical testing. And I'm not trying to advertise here. I'm not going to say who it is, but it made us basically be able to do this really quickly. Because I think you are in a better place if you can do the bioinformatics all by yourself. But if you need to analyze genomes quickly, we needed a pipeline that also good old cytogeneticists could work with, right? And people who had been in the field for a long time, who have all this knowledge, but who don't run the command line, who don't code. And that doesn't mean that they're bad geneticists, just means that they're not bioinformaticians. And so I know that, for example, Nijmegen is doing a different approach, but they have Christian Gillissen. If I had him, I would do the same. But currently, I try to hire him, by the way, Christian, if you're listening. So we did this. So I'm sometimes regretting it, and we are moving back to doing our bioinformatics ourselves. But this really gave us some kickstart. And I think now many labs in the world are in the same position when they're moving to long reads. The bioinformatics behind that is even more complicated. And so I think like a commercial version can help, but try to be independent, right? We are academics. Many of us here are academics. When I first started, what I really thought, I must say, and I actually started doing whole genome sequencing in 2014 with Christian Gillissen, I really thought that we're going to solve all our cases. I was very naive, right? But that being said, I must say, just also from the brief discussion I had just with Bill Newman before that, and with the work that I cited from Nikki Whiffen, this year and the last year have really been the most exciting for genome sequencing and non-coding, because we are finding so many mutations. Who would have thought that we find mutations in the small non-coding RNA causing intellectual disability at such high frequency. Right? This is unbelievable. It was there, like in our small cohort, we had several cases immediately. We just didn't understand it. And that's why I think if, don't be afraid, just go into genome sequencing and you will find interesting diagnoses. And I think with the right software at hand, you can do it quickly. And then I think this is my takeaway, and then I stop because it's 5. I hope many of you in the audience agree. Sharing is caring, right? We need to, rare disease is not something you can do on your own. We need to do this together and share data. We've just rolled this out Germany-wide, which has been difficult because of patient confidentiality, but we need to share our data in order to make better diagnoses for our patients and make it faster. And I think then we're going to find lots more things like the small non-coding RNAs. And with this, I think...
Malte, thank you so much. That was an absolute tour de force, incredibly stimulating, a wonderful way to start this webinar series. On behalf of the whole society, I want to thank you, not just for your work and your energy and your enthusiasm, but for what a role model you are to us all. Thank you so much for presenting. Thank you, everybody, for joining us today. As Malte said, I hope you join us next month to listen to Nikki Whiffen and continue to support this program. If you have any suggestions, we'll find a way of you being of speakers. We'll find a way of allowing you to flag those to us so that we can keep this seminar series very much reflecting what you want. Thank you all so much. Bye-bye.